All right, everybody, welcome. Welcome to the monthly CDCR seminar. It's June. A lot of people have graduated. We got our neurosurgery resident exam results today and they passed. Yay, 2 new neurosurgeons. It's good. And I'm really, really happy today to welcome 2 very special guests from the NRC in Ottawa, Dr. Kevin Henry and Dr. Martin Rossotti. We're so lucky to have Kevin and Martin visit us. They are longtime collaborators of SingLab and of the larger GBM Team Canada. That's a team of multidisciplinary scientists who are working together to generate new immunotherapies for glioblastoma. And in that capacity, I've gotten to know Kevin and Martin very well. We've worked together very closely. And again, the key thing is that although our expertises are very distinct, I'd like to call them complementary but non-overlapping. This has been a real joy to work with people with such amazing expertise, completely outside of my knowledge base, but so essential to advance anything that we're doing in studying GBM biology. So this kind of interdisciplinary mode of working together has been really, I've learned so much from both of them. And that's why it's such a huge pleasure to have them here and for all of you to meet them. And I'm sure that you're going to find them to be the same amazing resource that they have been to our work and all that their expertise has contributed. You know, I really wanted to share that incredible resource with everyone here at McMaster and certainly everyone in the CDCR. So I'm going to tell you a little bit about Kevin and Martin as I introduce them. So Dr. Kevin Henry is a research officer in Human Health Therapeutics Research Center at NRC in Canada, and he's also an adjunct professor in the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa. And prior to joining NRC in 2014, Kevin completed his BSc and PhD degrees at Simon Fraser University, as well as a master's degree at the London School of Hygiene and Tropical Medicine. And his work on the genetics and molecular immunology of antibodies and their interactions with antigens has resulted in more than 40 scientific publications and patents. And currently, Kevin is very interested in looking at biology and genetics of nanobodies, which you'll hear more about today, protein engineering strategies, integrating in vitro display technologies with high throughput DNA sequencing, and antibody engineering for the development of multi-specific therapies. And Dr. Martin Rossotti is a research officer and also in the Human Health Therapeutics Research Center at NRC. And prior to joining NRC in 2017, Dr. Rossotti completed his BSc and PhD degree at the Universidad de la República in Uruguay and a postdoctoral fellowship at the University of Ottawa. His research interests are also nanobody discovery, engineering, and therapy, especially the development of innovative selection and screening methodologies to identify high-quality nanobodies against difficult targets. He has authored more than 25 scientific publications and patents in this area. So the nanobody expertise in this room is now overwhelming. With that, I want to introduce to you Dr. Kevin Henry and Dr. Martin Rossotti.
Make sure I don't spill my coffee on myself here.
I completely forgot my instructions from Paul. I have a few announcements, but please go ahead. Well, I just have a few announcements. I forgot to make the announcements. I was so overwhelmed by introducing my friends. The pizza today was provided very generously by Drs. Henry and Rossotti. So this is an extra special rounds and perhaps it's garnered a better attendance than usual. I want to remind everyone that tomorrow, sorry for the holiday weekend coincidence of this matter, but tomorrow is the deadline for abstract submission for the CDCR annual research symposium. So please, guys, submit. We really want to see as many of you present as possible. This is a really exciting day. Our visiting keynote this year will be Professor John Bell. And, you know, for any of you to have an opportunity to present to Dr. Bell would be amazing. But yes, please do submit your abstracts. And the last thing is, I just wanted all of you to notice that I'm wearing some new swag. Have you guys seen my new swag? This is our CDCR lanyard, people. So we have a new lanyard now, and I'm going to be wearing, I know I don't have my ID attached to it, but Yes, all of you are going to get one. What do my instructions say? They are going to be at the exit. So please pick one of these up on the way out. Okay. So with that, sorry, Professor Hannon.
Okay. Okay, thank you, Sheila, for that excessively generous and totally undeserved introduction. I still have no idea how Sheila managed to do all the things she does, and she does it while maintaining an endless good humor and patience for all of her colleagues, even those of us in the federal government who can be mired in red tape. I also want to thank everyone from the CDCR and the broader McMaster community for welcoming us today. Can't imagine a more welcoming reception. We really impressed with everything we saw, from the facilities to the collaborative spirit of the center. It's so easy to get bogged down in science with experiments that don't work and negative funding decisions. But being here today, surrounded by all these brilliant people, especially the trainees and the early career researchers, one just has to feel, I guess, grateful and hopeful. So thank you all for that. So our group at NRC is interested in studying antibodies from various different perspectives, including the immunology and genetics, their structures and mechanisms of interaction with antigens, their engineering as proteins, and their use in diagnostic and therapeutic applications. So I figured I'd lead off here by touching on a few core concepts, I guess, and why I'm personally interested in these molecules. So I guess the first is that antibodies are not actually templated anywhere in the genome. So as you all recall, these are recombined from gene segments in each developing B cell. And the particular combination of gene segments that results in the non-templated nucleotides added between the segments serves as an indelible marker of each B cell that's born in our bone marrow. Unlike T cell receptors, of course, B cell receptors are further diversified somatically in the periphery following antigen encounter by somatic hypermutation. And this is targeted to the three hypervariable loops, and especially in the CDR3 loop, which serves as the dominant force in most antibody-antigen interactions, because it's where the gene segments come together and are joined approximately, and is the largest source of repertoire diversity in most antibodies. So together, these mechanisms serve to produce an amazing variety of different types of antibodies. And these can range from anything, including unusual cow antibodies, in which the conventional binding loops shown here don't actually do anything at all. And 100% of the binding is mediated by these very unusual cysteine-linked knob domains that are 5 or 10 kilodaltons that sort of protrude like a spear. Antibodies against viruses that have diverged by more than half of their nucleotide sequences from their germline form and have learned to sort of cross their arms to target the glycan shield. And even antibodies that have reached into the genome and inserted non-immunoglobulin proteins into their coding sequences, in this case, to broaden recognition of malarial parasites. Everyone will be familiar here with the structure shown on the left. This is the Y-shaped human immunoglobulin G molecule. This is one of the most famous structures in all of biology. One of the antibody systems we've worked on most extensively are single domain antibodies, which is the most inclusive name, I guess, for the variable domains of naturally occurring heavy chain only antibodies that arose in sharks in camelids and some cartilaginous fishes. These were both discovered in the 1990s. So the variable domains of these antibodies are called VHHs and VNARs respectively in those organisms. For today's purposes, we'll use nanobody, single domain antibody, and VHH pretty much interchangeably. So the important distinction I want you all to take away from this slide is that these molecules are distinguished from conventional antibodies by the fact that binding is mediated by a single variable domain on the heavy chain, rather than two variable domains that are non-covalently associated on different chains. So here we are in the original NRC labs on the banks of the Ottawa River, where there's been a group working on antibody fragments since about the 1980s. The camelid heavy chain only antibody system arose subsequent to the origin of the conventional heterotetrameric system in vertebrates by mutation of the consensus splicing site at the 5' end of the CH1 exon. And this results in direct fusion of the VHH domain to the hinge region. And because you lose the CH1 domain, which is critically required for pairing with the light chain, these antibodies, of course, no longer have any light chains at all. So the camelid VHH, in camelid VHHs, as shown, also known as nanobodies, the autonomous folding in the absence of paired light chain domain is made possible by the accrual over evolutionary time of a set of polar and charged residues. So in the picture on the left here, we have a nanobody in cyan with binding loops in white, and these residues are shown in various different colors, which replaced the hydrophobic residues, which were at the hydrophobic interface between these two domains in a conventional antibody. So that's what's shown on the right with the VH domain in cyan and the light chain in green and transparent. Our group and others have spent actually quite a bit of time trying to understand the basic science of how these molecules are formed. There are still a lot of unanswered questions here. So there's clearly a bifurcation in the humoral immune compartment of these animals, resulting in these two different types of antibodies. And it clearly occurs pretty early, such that the initial rearrangements of the VHH domains differ from rearrangements of the conventional VH system, with increased addition of non-templated nucleotides, which serve to produce longer CDR3 loops. We still don't actually know if these heavy chain only producing B cells go through an IgM phase. We can't detect any significant amount of heavy chain only IgM in serum. And I guess the other thing to note is that there seems to be additional mechanisms of repertoire diversification in the heavy chain only compartment. And these are manifested by elevated somatic mutation rates and isotype switching, which may actually occur in the absence of any antigen stimulation whatsoever. Most of these insights were gleaned through high throughput sequencing studies of the peripheral repertoire. We're also interested in the structural immunology of nanobodies and the mechanisms through which they interact with antigens. So it was noted quite early on in a few high-profile case studies that some of these antibodies at least like to target distinct sets of epitopes on proteins that are largely not accessible for the conventional antibody system. So what's shown in this picture is a superimposed set of conventional antibody Fab fragments on the left in complex with lysozyme. And on the right, we have the structures of a bunch of antibodies targeting the same protein. And what you can see here is that these antibodies tend to prefer recognizing the active site cleft, which is seemingly ignored by the conventional antibody system. So we and others have found through analyses of X-ray crystal structures that despite having paratopes that are roughly half the size of conventional antibodies, so again, there's only one variable domain, not two, and there's only 3 binding loops, not 6. The nanobody system seems to use similar types and numbers of non-covalent interactions to bind antigens, so hydrogen bonds, salt bridges, but they do so by concentrating these in a smaller region of space. And as a result, they vary similar solvent-exposed surface areas, but achieve slightly better shape complementarity of the interaction surfaces. This is an analysis we published in 2018. And one of the few good things that came out of the pandemic was an expansion of structural data on antibodies in public repositories. So the results of a current analysis of a larger data set are basically the same, showing that although there are fewer residues involved in the nanobody paratope, they tend to pack in more interactions per residue, such that your average nanobody epitope might be slightly deeper and more concave than a monoclonal antibody epitope. I should point out here, though, that there's a downside to the nanobody architecture, and what these antibodies are not very good at is binding small molecules, including short peptides, which is going to make what Martin will show you in a few moments even more impressive. So in the therapeutic space, the reason why most people are interested in nanobodies is their favorable properties compared with fragments derived from conventional antibodies, especially scFv fragments. Nanobodies have similar binding affinities as conventional antibodies, but as fragments, they're a whole lot more stable and more modular than scFvs, which can require extensive molecular engineering. So the other advantage here of any fragment-based therapy, including nanobody-based therapies, is that smaller molecular size tends to result in better tumor and tissue penetration. Although the downside of that is they tend to be cleared very quickly from the serum. So there are various strategies we can employ to improve their half-lives. Oh, yes. So I put this second slide in to make the same point. So I promise you it's not propaganda. I should have said at the outset, there is actually no patent now on the nanobody system. And when there was one, we didn't have it. So I've got no reason to lie to you. What's shown here on the left are regulatory approvals. As you can see in this graph, very few biologics that are antibody-based that have been approved are fragments. Those are shown in green here. And of those, only one was a nanobody, caplacizumab. But in applications that require a fragment, the nanobody system has pretty distinct advantages over scFvs. I don't have time to get into this too much, but the reason why is trying to get two domains to achieve correct packing in space and the successful recapitulation of the hydrophobic interface between the VH and VL domain in one amino acid chain, whereas in nature it was two. It's not straightforward. So the example shown here is CAR T therapy, where so far 1 of 6 approved products are nanobody-based. The other handy thing about antibody fragments, including nanobodies, is that they allow departure from the geometrical constraints of the conventional IgG molecule. So that's shown on the left here. And even if you take into account the flexibility of the hinge region, and even if you were to use most bispecific platforms, this is still limited to 2 of the same binding site, or maybe 2 different binding sites within about 50 to 150 angstroms in space. Whereas with the nanobody system, we can sort of tie these together like beads on a string to produce nearly any architecture. So I think I'm going to just try to illustrate this using a couple of examples. I feel like maybe this is the opening act for a rock star, so I don't want to overstay my welcome. So we'll see how many I have time for. I think I have a timer. We're doing all right. So the first example I'm going to show you is a campaign against a receptor tyrosine kinase called AXL. Some of you may be familiar with this target. It's being explored in a variety of solid tumors. There are a few important concerns and challenges with this target. First, that it's pretty broadly expressed. Second, that the extracellular domain is shed in AXL sync, and also that it has a pretty abundant high-affinity ligand. This hasn't dissuaded people from pursuing a variety of approaches, antibodies, ADCs, receptor traps, and small molecule inhibitors. So we went ahead and we generated some panels of monoclonal antibodies and single-domain antibodies against this target. They bound the first 2 immunoglobulin-like domains of the protein, and they fell into 3 epitope bins that are sort of grouped here as circles. Within each of these bins, the antibodies largely had similar affinities within a few fold. So when we took the nanobodies and reformatted them as FC fusions, so this gives you basically an IgG-like architecture, we found that they have basically similar ADC potency against AXL-expressing tumors as monoclonal antibodies. So just to take one example, we have an antibody in antibody FC in orange and a monoclonal antibody in red, targeting an overlapping epitope. We also found that any antibody targeting the site of the ligand interaction, so for example, the light blue line here, in the presence of the ligand is totally ineffective due to competition with the ligand, which was unfortunate but not unexpected. So what we decided to do is to see if we can improve upon what we had using the properties of nanobodies. So I'm just going to take a few moments to try to explain this slide in case anybody's unfamiliar with it. These are surface plasmon resonance sensograms showing the association and then the dissociation kinetics of these antibodies when they're flowed over an axial surface at different concentrations. And what you can see here is that if you take a monomeric nanobody, you get an association and then it comes off. Anytime you make a nanobody dimer, the dissociation is dramatically slowed, which is what we expect. And this is a result of avidity binding to usually two different axial molecules. But only a few of these nanobodies, the one I'll point out here is 6 and 8 with a short 15 residue linker, seem to saturate the surface very quickly, even at very low concentrations. So I'll just take the 2.5 nanomolar concentration here as an example. Whereas at similar concentrations of the constituent monomers or different dimers, you basically get hardly any response at all. And I'm not going to get into the rest of the story. I'm just going to say that the reason why is that this molecule is able to engage both arms simultaneously on one axial molecule. These sites are closely spaced within about 50 angstroms, and it's probably not possible for two Fab arms to engage simultaneously within that space. I'm not showing the animal study data, but this biparatopic antibody was also dramatically more potent in PDX models than any of the monomers as FC fusions, or than any of the dimers that can engage only on two different molecules. So when we produce these nanobody-based therapies, they're very small. Of course, they're cleared very quickly from circulation because they don't pass renal filtration. And they also don't have any of the effector function that are governed by the immunoglobulin FC, so for example, ADCC and CDC. So we need some additional strategies to add these. And there's a number of strategies we can explore. Some of the ones we use most often are fusion to nanobodies that target mediators of effector function like FC receptors and other host molecules. So just to briefly touch on this, as a strategy for half-life extension, one option is to take advantage of the neonatal FC receptor mediated recycling of albumin, which spares it from degradation in the lysosome. So the requirements here in terms of a serum albumin binding moiety were that it it binds cross-reactively to the albumins of humans as well as relevant animal models. It binds at endosomal pH, and it has to target a site that is distinct from where albumin interacts with FcRn. So to do that, we immunized an animal and generated an antibody library, and then selected it on the albumins of multiple species. And by sort of subjecting the panning outputs to high throughput sequencing, we were able to pretty accurately identify which clones were cross-reactive and which weren't. Yeah, interestingly, a few of these antibodies showed naturally pH-specific bindings. So it's actually a rare property of an antibody. So what I'm showing here is that there's one clone here called M75 that shows dramatically weaker binding affinity at endosomal pH, and this results from the protonation of several histidine side chains at the interface between domains 1 and domains 2, which results in a conformational shift in albumin. Here, what I'm showing is a second clone. It's called M79, which shows the opposite effect. So in this clone, endosomal pH produces results in protonation of His 367. which creates an additional hydrogen bond with the nanobody CDR3. And the formation of that bond you can detect using isothermal titration calorimetry. Anyway, so when we produce fusions between nanobodies that target antigens and serum albumin, these are able to bind to their respective targets, so binding to an antigen and binding to human serum albumin. What's shown on the right here is a co-engagement experiment showing that when we produce these molecules, they're able to bind to their targets, in this case, Clostridium difficile toxin A, and serum albumin at the same time when that's injected on top of the complex. And it doesn't really seem to matter which orientation we put them in. It seems to be generally tolerant to the fusion orientation and to however long the linker length is. So the end result of all of this is the extension of half-life from say half an hour for the monomer to a couple of days in a rat, which is equivalent to a couple of weeks in a person. What's shown in red here is the clone I mentioned to you that had a weaker binding at endosomal pH, and this gives an intermediate effect of half-life extension of several hours. We still don't fully understand the mechanism of this antibody. It does seem to involve, in addition to the pH sensitive factor, preventing binding to FcRn once the antibody is bound. This is kind of illustrated in the cartoon below these sensorgrams. It also seems to kick off FcRn once we inject it. So if we pre-complex FcRn to albumin and then flow antibody over that, FcRn is displaced. And we think there has to be some aspect of sort of conformational locking of this antibody that we're still trying to understand. We can also reverse engineer ADCC using nanobodies against natural killer cell surface targets. So this is just a diagram of ADCC. Here's a diagram of how this might work for a nanobody-based therapy. So as with the example of serum albumin, we find if we tether tumor-specific nanobodies to CD16 nanobodies, the binding of both of the arms is relatively the same when we produce them as monomers or in the dimeric format, as shown in these isoaffinity plots. And similarly, when we perform co-engagement experiments, the antibodies can bind both targets simultaneously. Just very briefly, I'll say that we haven't actually identified the target for the epitopes for these antibodies yet, but we have a pretty good guess at where they bind, and the reason why is that there's two isoforms of CD16, CD16A expressed on NK cells, CD16B expressed on neutrophils, and the nanobody I'm showing you here actually doesn't touch one of those isoforms, one of the allotypes of the CD16B isoform at all, but it does bind very well to the other. So those regions are shown here in red. It would be much more desirable to avoid the CD16B sync on neutrophils entirely. So that's what we're working on now. All I'll say here, I think, is that these fusion to a CD16 antibody gives you very potent ADCC in the picomolar range. And this works very well in multiple systems. So I think I'm going to leave it there. I hope I've at least convinced you that these molecules are interesting and potentially useful, and give a useful molecular engineering toolbox where we can easily modify things like affinity, valency, half-life, effector function, and molecular size. So at this point, I think I'll hand things over to Martine to continue the story here.
Okay, thank you for being here. And today I would like to show you one example about how is the chain of during the development of single domain antibodies. So, and in this case, there is a lot of attraction or these type of molecules are very attractive because we can rely on the specificity of the antibodies for targeting particularly some fingerprintings that makes different one protein, one receptor from a normal cell and also from a tumoral cell, because the efficacy of the therapy indeed is going to be based on the specificity of your antibodies to distinguish between the tumor and the normal cell to avoid any side effect. So one of the main challenges when they are planning to create these type of therapies is to identify the target. Because as we all know, most of the difference between a tumoral cell and a normal cells are very subtle. So this is the most challenging thing. And another thing is just we can create antibodies, but usually in order to trigger some therapeutic effect, we need or we request to those antibodies to have some high affinity. So with these two things in mind, the example that I am going to show you today is EGFR. Because EGFR, the particular thing of EGFR is that most of our cells express this receptor, but in some tumoral cells, there is a variant that differs from the natural in only one part. So for example, the truncated version that's supposed to be tumor associated have a truncation where this part is missing. So if we consider if this is missing, most of the tumoral associated antigen and the normal antigen is going to be not exactly the same, but very similar. So trying to find and developing an antibody to discriminate these type of things is going to be very challenging. But EGFRB3 is a good example because this deletion, it does remove, of course, remove a part of the protein, but also brings two parts of the protein together. So in this situation, it's something very particular because this junction creates like a neoepitope that is not present in normal cells. So if we are able to develop antibodies that are specifically targeting this, targeting and recognize this part of the receptor, we may be close to have something tumor specific. Okay. So in regard today, I'm going to talk about the EGFR, but EGFRB3, but this is nothing too novel. I mean, a lot of effort has been done in the past for a lot of scientists to try to develop this type of antibodies because they are very attractive. So there are a lot of reports where they describe the isolation and development of conventional antibodies, the one that Kevin showed in the past, that they have like really good affinity and also specificity. But so far, there is no report of single domain antibodies with this property. So based on the introduction that Kevin showed about all the good properties of the single domain antibodies, we wanted to exploit if we were able to achieve to develop these type of antibodies and use all the single domain properties in our advantage for many applications. So, and as I said before, those other groups, researchers, they were able to develop this type of antibodies, not simply, it was more complicated, but by the immunization of the EGFRB3. But a lot of people have tried to reproduce these kind of things in llamas. But when we perform this in llama, not only us, a lot of different groups, because a few years ago, EGFRB3 was like very hotspot, very attractive. So a lot of people was trying to develop this type of antibodies. But using the conventional strategy, what the outcome, yes, we were able to isolate really good antibodies with high affinity, but all of them were cross-reactive to both variants. So we are still, okay, we have something highly specific, but we are lacking of the selectivity. So in that case, with that in mind, we wanted to still pursue this, the development of these challenging antibodies. And this is another representation of what happened. When I said this is like the wild type, and this is like the truncated. As you can see here, this deletion brings together two parts of the protein. And in this case, this is like the neoepitope. So if we want to develop an antibody that is highly specific and is able to see these and detect these differences, ideally, we would like to develop an antibody that recognize both parts simultaneously, because we have to remember, it's clear that those parts are present in the wild type. So in order also, as this neoepitope is a small peptide, it's a short peptide, which is 13 amino acids. But as Kevin showed, the single domain antibodies are great for many applications, but one of the limitations that they have by how they are generated inside the animal is that they are not particularly specialized for recognizing linear epitopes. So that's why if we wanted to go ahead with this methodology, the peptide by itself, as it's very small because of how the immune system works, is not going to be immunogenic. So in that case, it's trying to pursue that. We conjugate the short peptide by using a carrier protein. And this carrier protein is going to improve or trigger an immune response. And we are hoping here that as a collateral, the animal is going to develop antibodies against the KLH, but also against the peptide. So if this happens, we will be in the good path. So once in this case, as Kevin talked, you can work with camels, alpaca, but we use with llamas. So once we have the antigen, the immunogen, we immunize the animal. The immunization schedule include like different doses at different times. Usually it takes like 1 to 2 months. And but the timeline is going to be very dependent on how immunogenic is your antigen. So that's why it's necessary to evaluate indeed the reactivity of the serum, the antibodies, against your peptide. And in this case, as we mentioned, for example, we immunize with a peptide conjugated to a carrier protein. So in order to test or to show that we have a specific immune response against the peptide, we need to evaluate the reactivity against the peptide, but in another context. So here you can see that the immune response was strong. So it's another sign to go ahead in the campaigns. And once we verify that the animal indeed generates antibodies, we go ahead with what we call, or to the methodology for isolating those antibodies that is called phase display. In our case, we use phase displays because it's the expertise of our team. But also, I like to mention that there exist many display technologies like ribosome display, yeast display, and many others. But today, I mean, my knowledge is about phase display, so that's what I am going to talk about. And just this is a general, because we said, oh, we work with llamas, but we work with llamas only at the level, we use the llama as a source of the material, the genetic material. I mean, we use the serum just to test if the animal indeed generated an immune response. But after that, the animal, we don't use it anymore. We just, from the blood, from the PBMCs, we have the B cells, and then we copy just the genetic material. And that material that we will include the sequence of the antibodies that were developed against your antigen, but also the amplification is specific for antibodies, but it's not as specific for the specificity of your pool of antibodies that you are cloning. So in this case, when you do that, you express antibodies in the surface of a virus. And something to consider that this procedure usually yield libraries that we call libraries to the population of antibodies from the repertoire of this animal. So, and after that, this is relatively not easy, but it's straightforward. But the success of your campaigns developing these antibodies is going to rely in a big part in how you design these selection strategies to isolate only from this high diversity of different antibodies, only the ones with the specificity that you want. So by the use of this technique, using this animal, we were able to isolate one antibody that was specific for the peptide. that here I am going to call it parental. And in this case, we evaluate the reactivity by ELISA and also by SPR. But we find out, that the antibody, this parental antibody, was able to recognize the peptide in the context of the receptor without recognizing the parental. But as you can see here, the affinity is very weak. It's like in the range, it's 40 micromolar, which is very weak. But at least we achieved one of the requirements, that was the high specificity for the EGFRB3, this tumor-associated variant. Because the most difficult part is to have something to work with. Once you have something, we have the methodologies and the knowledge to try to improve it. So this is what we did next. Okay, so something about the parental to try to understand if the antibody was worthy to pursue was to understand a little bit what is being recognized in the peptide. And as I said, as I showed you before, the peptide, this is like the wild type neopeptide. And then in order to know which amino acids are important for the recognition of the antibody, we perform something like it's called alanine scanning, that consists on changing every single position for an alanine. So if this position is important for the recognition of your antigen, the antibody is not going to be able, is not going to be able to recognize that. So, and in this case, we were able to show that there is the antibody at least show a high dependency for the C-terminal, and for the N-terminal. So in this part may explain why it's specific. Okay, so another, so as I said, the main limitation was the weak affinity. So now we are going to improve the affinity. And how we do that? So as Kevin mentioned, the recognition of your antigen is going to be mediated by the CDRs. So if we want to affinity improve, we have to modify these CDRs, because are the ones that have contact with your antigen. So there are many different methodologies. I'm going to be fast here, but one of these methodologies involves the randomization of some positions to try to create more diversity from this parental sequence. So in that case, it's very relatively easy to randomize to create brand new variants. Again, the situation is going to be having a methodology to isolate now from this second generation of antibodies, the best ones. So, and in this case, as I said, this stage involves like starting from all over again, but instead of using the llama, we start doing genetic manipulation without the involvement of the animal. So, and in this case, something different that we did this time and we did compare with other people was doing was, instead of using the peptide for selecting the best one, we used the whole receptor. Because the peptide is something completely artificial. In nature, the peptide in your cell, the peptide is not going to be as free as in the natural receptor. So by doing these methodologies and this improvement, as we perform the selection, we enrich, and then we identified the variants that were much better than the parental. For example, we optimize everything to the parental, so the parental looks like negative, but here you can see that there are some other variants, and we chose those variants to characterize. So the characterization step consisted on measuring again the affinity by SPR, and as you can see here, we got like an improvement from 100 to 500 times improvement on affinity. And something important is that we were able to increase the affinity against the tumor-associated version without compromising the specificity. So that was really great. So, and another thing, independent of this antibody, something that we always need to require or to request to any antibody that we are working with is to test the cell binding properties. Because most of the steps that we are doing during the characterization, the development, include the use of peptides or some recombinant proteins, but all those materials are not what the antibody is going to be seen in your cells, in your body. So this is something extremely important and it's a big requirement. And we perform this by flow cytometry or any other binding assay that involves cells. So in this case, we have 3 cell lines, for example, the U87, the parental that express very weak levels of EGFR. So as a consequence, it was necessary to use another cell line that overexpress the wild type or overexpress the B3. And as you can see here, this is a nanobody, one of the one that we select before. And you can see that this nanobody that recognize both with the same is not able to discriminate between the B3. But this is what happened with the brand new affinity variant. So I never told because I was waiting for here, but the first, what we call parental was not good enough because it was 40 micromolar affinity, but also lacked cell binding properties. That was the main reason to do all these affinity maturation campaigns. And the outcome, those are like the best of the outcome, but as you can see here, all of them were able to, in some extent, recognize specifically the B3 variant without recognizing the EGFR in the context of a cell, which is the one thing very important. Is fat? Do I need? I have 2 more. So in another, no, it's good. Okay. So in another, also, as we said that the cell binding properties is something extremely important, we'd like to test get the same information for different methodologies. So in another thing that we always like to do is, because the flow cytometry is, positive, negative, if you use this cell line, but indeed you don't know what is being recognized. So in this case, the second quality control that we perform is by immunoprecipitation. So we have those cell lines. We have the 3 different cell lines, and in this case, we solubilize the receptors that used to be in the membrane by using a detergent. So in this case, we are going to solubilize the complex proteins that are inside of the cell, not only the EGFR or EGFRB3. In this context, you will have the whole extract. So, and then you incubate with the different antibodies. And in this case, if your antibody is able to bind specifically and with high affinity, your antigen, it will be maintained in the resin. So in this case, after that, you just wash away everything that do not interact, analyze by SDS page. And if you have, you can identify this by mass spectrometry fingerprinting to know the identity of this protein. And in this case, as we already knew what we were working with, we use a secondary antibody that recognized by Western blot the EGFR. So in this case, as you can see here, we test here we have the control that I showed you before that cross-react with both variants, and you can see that the control is able to pull down the B3, but also the wild type. But in the case of the affinity mature antibodies, now you can see that there is a trace amount of the wild type, but mostly is the B3. So this is another step of proof validating like the high selectivity for the B3 variant. So, and for the last slide, it's just, we wanted to explore a little bit more and just to see what happened in another context that was measured the cytotoxic activities. Those antibodies were able to trigger that after recognizing EGFR or EGFRB3. So we did the same using the 3 cell lines. And to validate that the methodology, we use cetuximab because cetuximab is something that is available and is well studied, and it's known that recognize the wild type and the B3. So here you can see that there is response in both cell lines. But also we have a benchmark that was a monoclonal benchmark that was specific for the B3, and here you can see that only signal with the B3. So this is important because it's validating the methodology that we are going to use to show the potency of our antibodies. So when we did that, this is like the lead one. And as you can see here, the lead one was able to trigger the ADC activity only in the cells and that cell line that is overexpressing the EGFR. So this is good that is also able to recognize and also induce some functionality. And okay, so as a concluding remarks, I would like to say that first is single domain antibodies are great, but they have some limitations. And one of the limitations was that they are not specialized against a peptide to recognize peptide, small molecule, carbohydrate, or lipids. But in this case, we were able to isolate something with weak affinity, but indeed was useful because we use it to implement some affinity maturation methodologies to increase that affinity. And this affinity was good enough to recognize the native receptor on the cell, which is something essential for an antibody. After that, we show some functionality about those antibodies. I mean, they are not useless to be used by Western blot. I mean, you can use it for something else. And I don't want to convince you about EGFR-V3 as a target. The idea of this presentation was to show that sometimes during the development of the antibodies, you cannot achieve what you want. It's easy to say, I want an antibody with that specificity. Sometimes work, sometimes do not work. It depends on the complexity of this, of your antigen of interest. But at least all the methodology that we have been used here so far can be used and implemented for the development of this type of antibodies against other fingerprinting that are also tumor associated. Okay, thank you so much.
That was amazing. Thank you. Talk about efficient delivery of incredible information. I bet that was 4 or 5 or 6 years of work or something that you just summarized, but it's just, thank you so much for giving us so much insight into antibody design and development. You know, it makes me realize that we're lucky to have, you know, such tools. And I just want to remind everyone in my lab who works with antibodies from now on, a good musician never blames his instrument. Okay. So no blaming the antibodies anymore. You got to tweak things. Look how much work goes into developing these things that you guys use and complain about. So, I would like to open the floor to questions. Who has some questions for Dr. Henry and Dr. Rossotti? If there are no immediate questions, I have a question to start. Hong has a question though. Hong, you go first.
One of the redirection of our work is to globally define splice isoforms, but you can imagine the limitations we see is when we're looking for those ideal isoform-specific antibodies, very limited course. So it's just amazing to see your work. So one of my question is for because many of the splicing isoforms for that is occur not black and white. So in certain cell type, it's having a ratio of 2 different isoforms. In order to detection of those ratio like 30% or 50%, you suggesting to say if the antibody design is very specific, you can just using one isoform specific antibody to define the ratio, or it's better to using maybe combination of the 2 antibody, one is general and one is isoform specific to more define their isoforms regions.
You want to answer that, Martin? Or I
Come
can
and
give it a
stand
shot?
near the microphone. What a great question, Hong.
I'm going to give you my one sentence answer, and then he's going to give you the better answer. I honestly don't know. I think that all of that type of task, which is depending on specific antibodies to measure the relative expression of 2 different targets, is fundamentally limited by the fact that antibodies themselves are not directly comparable. So I guess between the 2 choices you mentioned, I would say probably you're better off using both antibodies and trying to make sense of the data combined, but I'm not sure it's necessarily going to give you the answer you want. And I'm not sure how I would get you the answer you want, but I'll think about it. All right, Martin, you're in.
Martin?
I'm okay. No, honestly, I have to be sincere. I don't know the answer. I am more an antibody design, but I leave to Kevin that knows more, and I think this answer may fit better to what you were looking for.
Yeah.
if it's specific, I can imagine could be part of what antibody is.
One can.
Is there a reason it has to do, has to be done using antibody, recognition, or are there other techniques that would be more suited, do you think?
There's already ways we can
Yes,
use sequencing to count it as, but the question is always in the
absolutely. Yeah.
how the real protein level is isoform different, so it's really valuable to have that isoform.
Yeah, because not always the fact, I think with the B3, what happened at the very beginning was that it was identified in many places. Sorry. Well, I have a good voice. That you always relied on having those antibodies to clearly show that the data that you are collecting by genetic analysis really correlate with the protein. That is not always the case because not always those type of antibodies exist. That's why it will be nice to develop those antibodies, but if they don't exist, it will limit your work. But if you find this type of variant, we can create the antibodies and try to start. I will answer your question with practical things.
There we go. Very nice. Any other? Eugen?
But have you ever, so like how would post-translation modifications like affect the ability of like antibodies to generate down the road? Like if you suppose that your protein of interest is hyperphosphorylated, but the recombinant protein I wouldn't think is in a hyperphosphorylated state. So how would you best, when you're designing things, like think about that?
That's a really good question. So I guess my number one answer would be, I'm glad you're asking, and I'm glad you're thinking about these things, because not everybody does. It depends a lot on what you're trying to achieve, right? Because I mean, some proteins, post-translational modifications are not really relevant, and there's a good chance you make an antibody against most parts of them, and it's not going to matter. But in other cases, it really does matter. And this is why sort of producing these molecules in the right state, whether it's phosphorylated or not, glycosylated or not, can have dramatic implications if you're trying to target a specific region. So I guess, my general answer would be, you got to know what you're working with. And, the way we can do that best, I think, is by having a clear hypothesis in mind about what we're trying to do with the antibody, and then making sure that we can check that before we start, which we try to do when we can. We can't always do it.
I think all these questions are illustrating why the interdisciplinary consultation is so important, right? Because Kevin can't directly answer some of these questions because it's dependent on the specific biology of the situation. And they're very adaptive people. Like they basically adapt their entire design approach to whatever they tailor it to your biological problem, which is why it's silly to think about, we actually order antibodies all the time from companies and we just throw them on cells and go, what's going to happen? That's fine for simple things like binding. But if you're thinking about therapeutics, I think it really requires this dialogue to be happening all the time, right? So is there another question? I think Tobias, yeah.
So how good is the technology to essentially put a payload sent for? So how good are they in targeting, for example, for like a radio isotope or whatever type of payload, because they mentioned in your talk that they don't have an effect, or that they can trigger effect on the mechanisms of their own. So how good is the technology to attach a payload?
The technology is the same. So, I mean, there's two answers. First of all, I think I probably skipped over the part where I would have said it's very easy to reformat these into a conventional geometry in which it's basically exactly the same. The technology is the same as using any conventional IgG. When and if you might be working with a different format, so, nanobodies as small little dimers and things, there's a variety of strategies. The technologies are not mechanistically different than for full-sized antibodies, but there are a variety of strategies to put chemically modifiable groups on these things. So, some of those are very straightforward, like adding a C-terminal cysteine, which sometimes works fine. Because the sequence is very short, it's often possible to modify it such that there's only one lysine left, so there's only one amine. There's other more complex strategies to site specifically add something. From the standpoint of chemotherapeutics and ADCs, I'm not really sure there's a huge advantage apart from potentially tissue penetration. There is an advantage, I think, theoretically for radiopharmaceuticals. And multiple people have sort of investigated this. And the reason why is that, the goal there is to sort of get in, get out, target something and be cleared. And there are multiple groups who have developed sort of chemical methodologies specifically geared towards that. Did that answer your question? Yes.
I have one last question. Kevin, when you were showing the campaign for AXL, you showed that one slide with all of the graphs that showed when you were developing the dimeric targeting approach. And by the way, that was a really helpful slide because you had the nice graphic of the antibody above every graph, which helped us understand what the results were showing. And I often wonder this because we, as you know, before we met you, we published a paper on the development of a bispecific Ephrin A2A3 targeting antibody against GBM. And we found the same finding as you. At the very end of that slide, you mentioned very quickly, oh, and by the way, the dimer has greater efficacy and binding than the two separate antibodies if they were added together. And we found the exact same thing with our comparisons. The bispecific antibody always worked better than a separate SCFV against Ephrin A2 or Ephrin A3. And I've always wondered, and we've never figured out why that is. Is it something to do with stoichiometry? Why would the dimeric approach work better?
There's a really long answer to that question, I think. So, I mean, it kind of depends, but I mean, in the example I was showing you, I hope I made it clear that the reason why it works really well is because you're getting binding of two arms onto one target molecule and it never ever comes off. There are other examples when the effect is actually mediated through binding and cross-linking of two different molecules. For example, resulting in clustering of receptors and their internalization and things of that nature. So in the case of your antibody, I don't think I know.
Neither do I.
I forgot one. The most important question is to thank to all the people that was involved. No,
Yeah,
I mean,
it's a big
there's a lot
team.
of people that
Yeah.
that has collaborated developing everything. So we are just here today showing that there is a lot of people behind.
So on that note, please help me thank Martin and Kevin for an amazing talk.